with Dr. Cory Tobin. Dr. Tobin, say hello.
Hello, everyone.
All right, so this is the first of a video series that I hope to be able to do. I'm in contact with a couple other biohackers in Los Angeles and a few other people in the community. I really wanted to give people a sense for what's going on out there and some of the people that are helping drive this, and Dr. Tobin's one of those. Set up a biohacker lab that we'll talk about in just a couple of seconds. But primarily, maybe just first wanted to start off talking about Dr. Tobin's background as an introduction for him. Got his bachelor's in biochemistry, moved on to get his PhD in plant genetics from Caltech, and then thereafter founded a biohacker group in LA. Previously, I think it was known as LA Biohackers. Yeah.
Well, I found out that while I was in grad school. It wasn't like afterwards.
Oh, really?
Okay. Like 6 years ago.
That explains a lot, because I'm going to get to a question on how you got your lab equipment, because I seem to remember some interesting stories along the lines of how you were able to build this lab and getting your lab equipment.
Right.
You guys are now called the lab, though, right? Not LA Biohackers?
Right. Yeah.
Okay.
The other name, the LA Biohackers name, proved to be very problematic in terms of like fundraising and like dealing with banks and the government offices.
Yeah, tell me about that. So did you sign up to be like a nonprofit and therefore had to do a bunch of registrations, or what type of government stuff did you run into?
Right. I mean, so first we started off as a C Corp and then applied to be a 501c3 through the IRS. And the IRS was actually the most like reasonable office I've ever talked to. They didn't seem to care like what I was doing. They was like, "Oh, you did your paperwork, perfect, good job."
That's
so,
great.
Like, you go into the bank to like open a bank account, and they're like, "What do you do? Are you breaking into computers? Or are you making like super bug weapons to like take out the planet?" I'm like, "No, man, we're just doing biology."
Who knew that our first line of defense would be the banks? That's fantastic to hear.
Well, apparently, as has been explained to me, after like 9/11, there's a lot more regulations on banks knowing who their customers are and what exactly they're doing.
Yeah, that makes a lot of sense. I like that. Now you've been working. Well, first of all, I've got a few questions for you regarding your schooling and kind of your decisions there. I have had a few people reach out to me on my website, basically asking like, "Well, how did you decide where to go? Did you decide to go into synthetic biology, etc., or a different route?" Because there's not a lot of places right now that really offer a degree in synthetic biology. It's still kind of a new buzzword out there, right? So you decided to go into plant genetics and you skipped a master's degree. And I know there's also some debate on that regarding if that's like a smart choice or when you would want to do a master's, when you wouldn't. Tell me a little bit about your decision-making behind going straight to a PhD.
Well, I mean, if you're going to do a PhD in the sciences, most of the time you don't do a master's first, at least in the United States.
Okay.
You just go straight from bachelor's to a PhD. The masters tend to be for people who are like getting professional degrees or like we're working for a company and want to go back and get more training in a different field.
Interesting.
So I was at California Lutheran University in Thousand Oaks, California, which has like no research going on. It was just basically a teaching school. And I had like this scholarship from Amgen that was paying for part of my tuition. And so my thought was, okay, Amgen, their headquarters is in Thousand Oaks. I'm going to work for Amgen after school, after I get out of college. And then like my senior year of college, which was 2007, I pull up some news articles and realize, hey, Amgen's laying off like a quarter of their research staff. I'm like, oh, I guess I'm not going to work for them anymore.
Yeah.
So then I started figuring out what I should do. One of my advisors was just like, hey, why don't you apply for grad school? Because like this entire time I'd been doing like research programs during the summer. I'd been actually like interning at Caltech while I was living in Thousand Oaks and like taking a train out to Pasadena, where Caltech is in the summer times, to like do research over there. And I was just doing it for fun. But it turned out to be pretty helpful for getting into Caltech for grad school. And so I had one of my advisors write a letter of recommendation for me and got in there. And I never really like this wasn't my life's plan to become like an academic. It was just like, oh, I guess I can't work for Amgen. Maybe I'll just do something else.
Do something else. Hey, at least Amgen paid part of your way, though, it sounds like, huh?
Thanks, Amgen.
Plug for them. Now they're getting some advertising for it. So what interested you in plant genetics? Like, why go that direction?
So my first year at Caltech, I did a bunch of rotations. So you spend time in different labs for about a year. And one of the labs I worked in was this plant genetics lab. And I really enjoyed it. I liked the plants. I enjoyed working with them because there wasn't like this constant need to, it wasn't like you're adopting a child. Like if you go into like mammalian research, you're working with mammalian tissue culture. It's like you've adopted a child and you have to be in there like every day changing the media.
Really?
And I got so pissed off like having to just like baby these cells all the time.
Now is that just because like with the plant you can just like the plant in vivo is going to be fine in a lab, but working with mammalian genetics, usually you're still working in a petri dish, and so you have to be concerned about like the growth and the media involved there, or is that the story?
Right, yeah. So if you're doing in vitro stuff, you've got to be like constantly getting them growth media. If you're working with like flies, you have to be like constantly propagating. Like you make a transgenic fly, you have to basically keep growing it as long as you want it.
Huh.
With plants, you just like put some seed, collect the seeds, toss it in the box, and you can come back to it later.
Cool.
And the same thing applies for other things. Like working with mice, if you make a transgenic mouse line, you got to keep growing your mice, otherwise you lose it. Whereas I felt like plants, I had like this huge arsenal in front of me. Like I had thousands and thousands of different genetic modifications to these plants, all sorts of different SNPs, different inserts that are available. They're sitting in like boxes on shelves. I can like pull up any. That's the box right there. Pull it out, get the seeds out,
and just give it a shot.
Yeah.
Huh. Very cool.
So I really like that.
So how far along in your process of plant genetics when did you become interested in synthetic biology, or when you were initially getting into plant genetics in the first place, were you already interested in synthetic biology? Because my guess is you can do plant genetics without having to mess with their genomes at all, right?
Mm-hmm. Yeah, you can. Like a lot of people have been doing plant genetics for
a long time,
before we even knew what DNA was, or the DNA carried the genetic information. Mm-hmm. Like Mendel didn't know shit about DNA when he was doing plant genetics.
Right. That's a good point.
I actually got interested in synthetic biology while I was an undergrad. I tried to start an iGEM team at my university. It didn't go out. It never happened. Just like trying to get university administrators to allocate money for some stupid kids to like play biology or whatever.
Tough to do.
Yeah, it didn't work out. But so I guess throughout my career as a grad student, I never really did like synthetic biology. Like I made plenty of transgenic plant lines, but that's not really, I wasn't really doing it. The purpose of like building something new was more for just understanding how plants work, which I don't think actually counts as synthetic biology depending on your definition.
Okay.
But I always go to like the DIYbio conferences, so it's pretty cool.
Yeah. In fact, and you recently spoke at a conference just last month, right? September, Biohack the Planet?
Right, Biohack the Planet. Nice. As a great conference. That's
good. I've got a few questions coming on that one. I've never been to a conference quite of that nature, so I'm interested to hear how the process was. But before we get to that, a couple other questions for you. So you started the lab while you were still in college. When did you kind of have the idea that you wanted to bring in other people, help collaborate with other people, and really mentor a lot of new people?
Right. So I wasn't even like the first one in the group. It was a bunch of people around LA who were just like meeting at coffee shops, and just talking about biology. And eventually, at some point, our group kind of got big enough. We decided, hey, let's try to find a space. And so one of the people in the group, Tor, was a member of Null Space Lab, which is a hacker space in downtown LA, and they let us use a corner of their hacker space for like a year. So we kind of moved in there and just kind of kept growing. Like I've never had like this thought, like let's create a hacker space for biology. It was kind of like it just happened.
Okay.
The a bunch of people, and they're all kind of like trying to work on projects, and just kind of like spiraled from there.
Very cool. Now, I seem to recall that you had an interesting story on how you got some of your equipment when maybe you were launching your own space. You want to tell me a little bit about that?
Yeah, so I mean, a lot of this stuff is just comes from like universities dumping equipment. Like someone will say, "Hey, I saw UCLA's like trashing a bunch of stuff back by their dumpsters. You should come get it." And so I'll also like get a truck and just drive over there.
That's
and
great.
Load it up. Yeah, and like we have some hoods that people were like, they probably had some sort of nefarious business going on that they needed have a laboratory for, and I guess they were going clean and want to get rid of their laboratory and gave us some other stuff.
That's fantastic.
That's all I'll say about that one.
I don't even want to know how you made those contacts, but that is hilarious. You're probably one of the first like nonprofit that was funded in part by collecting materials from illicit drug trades. All right, so in terms of the biohacking community there, I've been curious a little bit because obviously California is a little bit more of a hotbed for the biohacking community. And then you've got some places back east where you've got a bunch of labs that are in close proximity to one another. So do you guys ever collaborate? Do you have a good sense for what other biohacking labs are doing? Or do you guys have meetups that are joint between you guys? What do you do there?
I mean, in general, I know what everyone's doing. Everyone's got Twitter feeds that I follow.
Okay.
In terms of communication, like back and forth communication, a lot of the leaders of the biohacker spaces have a mailing list that we talk on occasionally. And once in a blue moon, when we actually meet up in person, like at Biohack the Planet, we have some good conversations. But other than that, we're not like constantly daily emailing
not
each
doing
other.
Much. Yeah, sure. Makes sense. Now maybe to get into a little bit of your personal interests, and this will segue into your speech that you gave at Biohack the Planet. I found an article that was written at makescene.com where you kind of explained an interest that's, I guess, captivated you for the last year, couple years or so.
Oh, the one by David Lang?
Yeah, well, this was about the nitrogenase.
Yeah.
So
that was actually a project that kind of like got me interested in meeting up with these people in LA, which is one of like the founding projects we had going on at LA Biohackers in the early days.
Okay.
I had this idea for this project, and I was kind of working on it on my balcony at my apartment. And so I was looking for other people to collaborate with, or there's a space to work in, because I didn't want to work on it at Caltech, because at the time I had intentions of like commercializing the technology. And
right. If you do it at their lab, it's probably partially theirs, huh?
Right. So I always made a point to like not even use email, like not use my Caltech email address for anything related to that. Just like make sure it's clear that I wasn't doing any of this on university property.
Okay.
So I was doing it in my apartment, and then I met up with other guys in LA, guys and gals, and that was kind of the very beginnings of LA Biohackers in like 2010, 2011.
So let's zoom out. What's the master plan here? What was the goal? What were you trying to do?
So the goal was to make a nitrogenase enzyme system that is impervious to oxygen. So most nitrogenases, which are enzymes that convert nitrogen from the atmosphere into ammonia, which plants and other organisms can actually use, the nitrogenase is the enzyme that does that reaction, and it tends to be, those enzymes tend to be poisoned by oxygen, and so they are all bacterial enzymes, and these bacteria form symbiotic relationships with plants, and they live in the root nodules. And the root nodule provides like this kind of almost anaerobic environment for bacteria to work in. And so people have been trying to introduce nitrogenases into plants for the longest time, because if you could do that, you can make plants that don't really need any fertilizer. You still provide them like potassium and phosphorus, but one of the major components, the nitrogen, would basically come as a freebie if you could like put that gene or the genes into the plant.
Okay.
If you just try to splice that gene into a plant, or like that set of genes into a plant, it's not going to work, because plants produce oxygen, and that interferes with the nitrogenase enzyme.
Right.
So there had been a bunch of literature on this bacteria in the 90s in a lab in Germany about this bacteria that fixed nitrogen in the presence of oxygen. And so around like 2001 was the last time there was any publication on it. And so I thought, well, I should go investigate this. Like, even though there's like a good chance that it's bogus because no one else has been working on it, so someone else who has more information than I do decided not to work on it, it's probably a bad idea, but I still want to do it.
Right.
And so I tried to get government funding for it, and everyone's like, fuck off. This project's stupid. And they're in the end, they were actually right. But I still did it anyways, and went and got soil samples from Germany and isolated the bacteria and grew them up. We ended up forming this big collaboration with Harvard, Imperial College London, Michigan State University, and a couple other universities. Because I had been like publishing all my results on my wiki, and they were just like reading it. And one day when I published my like results showing that I like isolated the bacteria that had been previously published, all of a sudden I started getting emails from around the world being like, "Hey, can I collaborate?"
Wow.
Like, oh, okay, sweet. You got money? So
these are like grad school people and like leaders. Yeah. Are these like principal investigators in other places? And
no, it was like postdocs. Actually, I think James Murray was a lecturer at Imperial College London, which I'm not sure what that actually is, if that makes him a PI or not.
Okay.
I'm not sure how their university like hierarchy
system
goes.
Works. Yeah. So the basic idea was essentially this then, right? So like the atmosphere is something like what, 70% nitrogen, right?
Yeah.
So most of what we breathe in is not necessarily straight oxygen, but unfortunately it exists in a diatomic molecule. So you've got the two nitrogens together. You're looking for something that could break down nitrogen from its diatomic pure element that you'd find in nature, so that a plant could use it and therefore wouldn't need that component anymore from fertilizer in order to help grow, right?
Right.
Okay.
Yeah.
So you go to Germany, you get some...
I didn't go to Germany. Okay, so this is like another interesting side story. So one of the guys at LA Biohackers, Dirk Newman, he's a German dude. His dad had a friend in Germany who like happened to work at this place where the sample had actually been collected the first time. These bacteria grow in the topsoil of charcoal piles. So when you make charcoal, you like put wood, you like make a big pile of wood, and you cover it with dirt, and you light it on fire, and the wood pyrolyzes, releases all these toxic gases, and the bacteria were originally discovered growing in the topsoil on top of one of these like burning charcoal piles. Okay. And so Dirk's dad's friend was the owner and proprietor of a charcoal factory in Germany that happened to be where this bacteria was originally found. So managed to get some soil samples.
That's insane.
From there.
What are the odds?
I know, it's crazy.
So you and I think your talk was on this at Biohack the Planet, but it seems like things didn't quite work out the way that you wanted to. So maybe give us an idea of the results.
So we ended up like sequencing the genome, figuring out like first off confirming that it was the exact bacteria that had been previously published, because we're not working with we didn't like get a sample of the bacteria from the original scientists. We're like re-isolating it from the environment. So first we confirmed that this actually was the exact species that had been previously reported on. Figured out some interesting things about it. Has a really weird genome, and it's probably not. So originally it was classified as Streptomyces, but probably not Streptomyces. Probably Actinomyces or something else. But anyways, that's boring. Interesting part was that we determined that it does not fix nitrogen.
No, that's too bad.
I guess the good thing is that no one else is going to waste your time on it, because we published it, so now everyone can see that this thing does not actually fix nitrogen. And we went to great lengths to make sure that we did this thing right, and that the results are not just like some artifact. It really does not fix nitrogen.
And you'd said you'd published some material regarding this, right? Is this material that you were just publishing on a wiki, or did you end up doing something that's
we ended up doing a paper. It's in Nature Scientific Reports.
Very
it's
cool.
Open access. If you just Google my name and like Nature Scientific Reports, you'll probably find it.
That's pretty awesome. And I could be wrong about this, but my guess is not a lot of biohacker spaces publish in academic journals. Would I be wrong there?
Oh, that's true. Yeah. And actually, I wouldn't have chosen, like, if it was just me doing it, I wouldn't have even gone to that length. I would put it in, like, Bioarchive,
Okay.
which is a open access, it's called a preprint server. So it's not actually peer reviewed.
Mm.
You can just submit your manuscripts to this server. It's very common in physics, chemistry, math world. Just publish your like
Some
preprint
of your
manuscript.
findings. Okay.
For some reason, biologists have adopted this like crazy corporate controlled system of like publishing in these journals, and there's like varying levels of prestige associated with each one. And so because my project ended up being this huge collaboration that involved people who were climbing the academic ladder, they needed to have some actual
Got it.
like university
Let's
approved.
have some clout, right? Yeah.
Which is kind of a pain in the ass because everyone wants to like shoot for Nature first or Nature Science Cell, and then get shot down and go to the next level, and they get shot down and go to the next level. And so it's kind of annoying. I was just like, just publish it, guys. I just want to get it out there. They're like, no, we got to do it because I need to get tenured. I'm like,
that's
okay.
Crazy though. Well, I guess that's the nice thing is now you're published, which is fantastic because if you decided you wanted to go a little bit more the academia route, you could do that.
Yeah.
Speaking of which, you kind of have yourself listed right now as an independent scientist, I believe is what I had read somewhere. So
yeah.
Tell me what that means.
LinkedIn profile says indie scientist.
Yeah. What does that entail?
Doing science without any money, I guess.
Sounds like a dream.
I know it is.
That's fantastic. So you help out a lot of times people who are just getting started. You spend a lot of your time with the biohacker space that you created. What type of projects are other people working on there, and what levels of competency do you get coming to these?
So we get everything from like junior high students who are working on a science fair project up to people who have PhDs, postdocs, whatever, who are like trying to start a company. Maybe they don't have like a bunch of funding yet, but they just want to test an idea out, and so they're kind of doing it from the side. Some of them are people who are like currently working as postdocs at university and doing like a side project. Other people are doing it full time. Other people are just like interested non-professional biologists.
Okay. When you say full time, you're meaning like full time, like they're there in the lab day in day out type thing.
Yeah, we have a couple people who this is like the thing they're doing. They quit their job and are like trying to make a project work and eventually commercialize it.
Wow, that'd be really interesting.
So I guess.
Go ahead.
In that sense, we're kind of like a biotech incubator, but I'm not really like pushing that aspect of it too hard. But we do have some companies which I guess are incubating in our space.
That's pretty cool, though. It's pretty cool. Love to talk to them sometime, maybe about what it is. I would imagine if they're doing some of the incubation, or if they're trying to start this company beforehand, is it something where they're nervous about letting their idea get out because they think some bigger company is going to go for it? Or would it be like, no big company is going to do this, which is why it hasn't been done, which is why I'm here in a biohacker lab trying to get things started?
I mean, people are always skittish about giving away information, especially when it's like early stage, non-patented stuff. I think the companies that are working in our space are probably would fall in that category. I think they realize that it'd be very easy if someone actually thought their idea was a good idea, they could scoop it up and do it faster than they could.
Yeah, more resources.
I think those thoughts are usually unfounded. Like most of the time, companies that have a lot of resources that could pull that off are not interested in your crappy idea.
Yeah.
You got crappy ideas to follow.
Yeah. I've found that with just entrepreneurship in general. My family does a lot of different businesses, and that's kind of been my experience, is that a company gets really good at doing a certain thing, and that's pretty much all they're really interested in doing. Lots of times they're not, which is why sometimes you see some companies come up and disrupt the system, right? Because you've got Netflix that comes along and helps wipe out, Blockbuster Video and Hollywood Video and stuff like that, and the other guys didn't even want to do it. They got on the train way too late and essentially got submerged by the new kid on the block. So makes sense. It's so a couple other questions for you. with regards to maybe your next interests now, where do you go from here? Is this something where you're still kind of hunting down how to solve this nitrogenase problem, or that's just like on the back burner, you're doing other stuff?
So like the nitrogen project, it's pretty much over. Like I'm not working on it anymore. I think all the collaborators in that project have pretty much dropped it at this point. Mm-hmm. Just because they've already invested like years into this thing, mm-hmm. and there's nothing else to really do. Mm-hmm. I mean, there were some interesting aspects to this bacteria, mm-hmm. but everyone was really counting on it being able to fix nitrogen because that was where the big money was.
Yeah.
So I think everyone's kind of moved on and trying to hit their home run somewhere else.
Makes sense. So what other type of things are you interested in then? What's keeping you intrigued there at the lab?
So me personally, I guess what I'm working on now is just like exploring other fields that I've never had the opportunity to explore before. Like right now I'm really into microfluidics. So cool. I'm just like messing around with stuff. I'm kind of like 10 years behind on the technology. And so I'm just kind of like trying to figure out like how to actually like make like a very basic microfluidic device. I guess now that I'm no longer a grad student anymore, I'm just kind of exploring whatever the hell I feel like.
Yeah. Well, that can be powerful. I mean, you get a strong background. I think that I had seen that you had done some bioinformatics in the past. You had some experience there. Is that right?
Yeah. So when I was as an undergrad at CLU, during the summer times, I was working at Caltech in Barbara Wold's lab. And so I was like a self-taught programmer. I never took any never did a CS degree or anything like that. Mm-hmm. My degree at CLU was in biology. Mm-hmm. And so I guess at that time, bioinformaticians were not that
big of a thing.
Frequently found.
Okay.
This was like 2005, 2006. And so she had this prototype, Barbara Wold had this prototype of the Selexa sequencer, which eventually got bought out by Illumina and is now called like the Illumina HiSeq.
Okay.
And at that time, it was so Selexa was like a really small company and had this device, trying to make something happen. And so Barbara's lab was working on both like the wet end of it, like figuring out how do you actually sequence DNA with the DNA sequencer, and how do you analyze data on the back end. Because at the core of it, this machine just takes pictures of like this glass slide. And you take those pictures and turn it into an actual like raw text file that has A's, T's, G's, and C's in it. And all the software was like a total mess. And it was like, if anyone's familiar with like compiling software, like you actually executed the software stack by like running a make file, and it like executed a bunch of like Java and Perl scripts, and like it somehow kind of worked.
Wow.
It was like, so she had kind of hired me on, or let me work as an intern on this project to try to make this machine actually consistently put out A's, T's, G's, and C's after you run DNA through it. And so I'm guessing a bunch of my software is hidden away now in Illumina's software stack.
Man, Illumina's made a killing. They made a lot of money. It's too bad you weren't in a little bit on some of that cash, huh?
I bought stock.
That's smart. That's good.
When I first saw it, I'm like, oh, this technology is badass. I'm gonna get in on it. So I bought some Illumina stock, and it turned out to be worth a lot of money later on.
Yeah, smart move.
Yeah.
Maybe that leads me to my next question, which is like just in the field of biology in general, what's out there that you're seeing that you feel like is cutting edge and really cool that's piqued your interest?
Biotech. Let's see. There's a lot of cool DNA synthesis companies out there. They're trying to bring the cost of synthesis down, which I think is a big limiting factor in synthetic biology. Mm-hmm. There's a lot of there's like tons of different companies out there solving different diseases, and so that's always cool to see. I guess I'm always partial towards the DNA sequencing and DNA synthesis companies. So I've been playing around with like a nano, what's it called? Oxford Nanopore Minion. I think it's pretty badass. I haven't really got to work yet, but
What does it do? What's the idea behind it?
it's a sequencer. It's like this little tiny sequencer that plugs into the USB port on your computer. And you
Wow.
just like samples onto it. I think it has a lot of potential, and it could be a really killer device. So far, I haven't had very much good luck with it, but I wish Oxford Nanopore all the best luck.
Yeah.
They got some cool technology. I think Illumina is going to assume out of existence, but there's I noticed a lot of like cool projects coming out of like iGEM teams. Like they come up with an interesting summer project, and eventually turns into a company, which I think is pretty awesome. Because most of the time, biotech, or at least historically, biotech has been like older people, like grad students, postdocs, starting a company out of university. But it's really interesting to see a lot of like younger biologists starting biotech companies and actually getting funding for it. If you look at like DIYbio's incoming classes, a lot of it is college students and like early grad students. So it's really interesting to see how the demographics of biotech startups are changing.
Yeah, there was a few that I followed for a while now, kind of dropped off the radar. One of them I was interested in was the bioluminescence company that did the glow in the dark plants, right? Are you familiar with that one?
Yeah.
Yeah, I was pumped for that one. I did a huge blog post on my website to try to figure out how bright are these plants and how far along are they in terms of, bringing the product to market, etc. And it seems like they're still kind of plugging away, but it's unfortunately, as many people find out when they start to get into messing with genomes, the metabolism is messy, right? And so things just don't go as smoothly as you would like them to necessarily. Which brings me to maybe another thing that I've seen recently that's kind of intriguing, and that's people messing around with creation of different biological products outside of the cell by just using enzymes. I think there was an article in the Atlantic just the other day where they talked about freeze drying enzymes, putting them on some paper that essentially you could add some water, and they would help to create some medicines so that you could ship these all around the world and wouldn't have to worry about quite as much difficulty in terms of working with a cell to be able to produce these. So that was kind of interesting. Another company I've been interested in as of late is Organovo. Have you heard of Organovo? They're in your neck of the woods.
Yeah.
Yeah, they're an intriguing one. I would love to see the type of stuff that they could come out with, especially once people start to do some biohacking. And because I know that one of the big difficulties, which maybe brings me to one of my last few questions for you. You guys use CRISPR there when you're working with stuff, or are you using viral vectors? Like how is it that you're doing some of your genetic engineering?
And there's one person at the lab who's using CRISPR. He's sort of using it as like an introduction for himself into the world of biology.
Okay.
I don't personally use it just because I never had a need for it.
Okay.
I know it's like all the rage, and so I should probably get on board and like do CRISPR just for the hell of it.
Sure.
But a lot of times you don't even need CRISPR. You only need it if you're going to make a very specific change to an existing genome. Like if you want to go into a plant and like change one base pair at a very specific point. That's a case for CRISPR. But a lot of times you just like want to insert a gene or a set of genes into a plant or a bacteria. You don't even need it.
Really?
And it's like more work than it's worth.
Interesting. Very interesting. So maybe my last question for you, actually, I was going to ask you about Biohack the Planet. Tell me a little bit about what that conference was like. How was it being a speaker there?
It was a lot of fun. It was probably one of the most fun conferences I've been to. It was definitely much more casual. There wasn't really any like formal guidelines or rules for giving talks. There was people showing videos. There's one guy who came on stage wearing a fish costume.
Nice.
There's plenty of drinking and socializing.
Sounds like a party.
Yeah. And a lot of people chanting "fuck the system."
That's hilarious.
Which you'd never see at like a regular like most like society conferences
Right.
or like a Keystone conference or whatever, you would never have like people like yelling from the audience at the speakers.
Yeah, well, my guess is that alcohol there probably had something to do with that.
Yeah, that's right. And a lot of the attendees weren't your traditional scientists.
Mhm.
There was a lot of, I guess, biohackers, people from other biohacker spaces around the country, and then a bunch of like random folks who were just showing up to see what it was all about.
Cool.
Which you don't usually find at like a society conference.
Mm-hmm.
Like if I went to, so when I was a grad student, I always went to the Arabidopsis conferences, and there wasn't even like reporters there. It was just a bunch of scientists.
Straight academics.
Whereas in this case, there was like reporters there. There was all sorts of people there just showing up to see what it was all about.
That's kind of cool. How many attendees do you think were there if you had to ballpark it?
80, maybe.
80 or so? Okay. And that's just the attendees, or does that include like the speakers and the presenters?
That probably includes everyone. Yeah.
Okay. Very cool.
So it's definitely on the small side in terms of conferences. But.
Well, I'd heard about it before, but my guess is that it's still early stages. Is this wasn't their inaugural event? They've had a couple, or this is the first one? Okay. Good deal. All right. So last question for you would be, is there a question that you wish that I would have asked, or something that you would have wanted to talk about that we didn't get to?
Oh. Well, I guess one of the biggest things these days that I'm working on is like getting students involved in biology. And so I guess it's more of a question that I have for the rest of the world. It's like, what's the best way to get students involved in science and how to scale it?
Mm-hmm.
Like right now, what I'm doing is like tutoring people one-on-one. Like that's how I make my money. That's my job now, is like tutoring high school students with their science fair projects.
Mm-hmm.
And so like I can get them involved in synthetic biology or any kind of biology in like a one-on-one situation. But I have to figure out a good way to like scale this.
Mm-hmm.
Like how do you do laboratory education?
Yeah.
It's very different from like textbook education.
Uh-huh.
I read all the textbooks as an undergrad and like pretty much like memorized them, but that didn't help me when I went to grad school. Like the only way to learn how to do research is to get dropped in the deep end and just figure out how to swim.
Interesting.
And I don't know how to scale that to like mass education.
Yeah. It reminds me of something that's not quite along the same line, but to some degree it is. There's a company, I think they're out of Sweden, and they're doing virtual labs. And I had an opportunity for just a couple minutes to play around with one. So it was a Samsung VR, right? Throw it in the Oculus or whatever, and you could go and perform a few things. What was nice about it is you could zoom in and it would explain things about the DNA and what a PCR machine was doing, etc. So that was nice because I've read a lot about biology, but I've got to be honest, like if someone dropped me into a lab and I had to follow some protocol, I would be toast. There's nothing, I would have no idea what in the world I was doing,
Yeah.
And it sounds
there's
like
no way to like learn it without actually doing it.
Yeah.
And to learn it really good, you have to not just be given a protocol, but to do, like everyone as an undergrad, you take biology lab courses, chemistry lab courses.
Right.
And you walk in to the beginning of the class and they give you a manual and it has all the steps of what you're going to do, why you're doing it, what you're supposed to see.
Yeah.
It's kind of like fill in the worksheet and then you get your answer.
Mm-hmm.
But once you start doing your own research, you have to figure out how to like formulate a question and then how to actually figure out how to like test it.
Yeah, the best way to
go. What kind of protocols you need to use. Okay, so now you figured out you have to do like a mini prep to do this one step. Well, what's that? What do I have to buy? How do I do it?
Right.
And whenever you're in like a formal learning, like a school or whatever, you're given a lot of stuff up front. You have to figure it out yourself.
Mm-hmm. And
the only way I've figured out how to get people to really learn how to do research is just like make them learn the hard way.
Right.
So like here's a project, figure out how to do it.
Yeah. That makes sense. And I mean, obviously it's the way that you do it, 'cause it's the way that you had to learn, and which is nice because I think most people don't want to do that, right? Like if we showed up to our biology labs and our chemistry labs and we had to figure out what to do, like people would probably be there for hours and making mistakes, and they got places to go, and so they don't necessarily want to do that type of thing, but they need to.
I think what makes it harder is that you can actually hurt yourself in the process if you do it wrong.
That's a good point.
Laboratories are not safe places. Like, if you're going to learn how to do programming, you can just like open up a book, or like pull up your compiler, open a text editor, and start going at it. And if you mess it up, it's like nothing really happens.
Yeah.
It doesn't compile. Yeah. But if you mess something up in the lab, you can cut your fingers off, you can catch yourself on fire.
Yeah.
All bad things can happen. And so there's like a fine line between dropping people in the deep end by themselves and like watching over their shoulder and like telling them what to do.
Have you had any close calls at your lab? People doing some crazy stuff where you're like, whoa, let's slow down on that?
I mean, so like when people first want like approach me and say, hey, I want to do a project in the lab. If they have a lot of experience, I say go for it. Like you've got 10 years of university experience, just like go for it. I trust you. Mm-hmm. With a high school student coming in who's never touched a pipette before, mm-hmm. I either try to mentor them myself or pawn them off on a different tutor who can actually work with them in the lab all the time. Mm-hmm. And so we haven't really had any safety issues. I think we've had cases where people are like about to do something bad, but since I was there or someone else was there like watching, I'm like, hey, put that down. You're going to blow yourself up.
Right. With regards to getting younger people involved in synthetic biology and genetic engineering, just in general, how do you see the future of this industry kind of evolving? I asked this because here's a little bit of background. I spoke to 3 professors at a local university regarding synthetic biology, and I got 3 wildly different answers. One of them was like, synthetic biology, I wouldn't touch it with a 10-foot pole. Like, it's going to take forever to commercialize. If you went into this area, you'd graduate and have nothing to do and a lot of debt. Someone else was like, you could do it, but the smartest thing would be to go into business, because the scientists never make the decisions at a company. It's always the business people that are making the decisions. So if you want control, go be a business person. And then the third person was like, those people are all crazy. Do something unique and start a business on it, because it's the scientists that can do that primary research and go and launch something as a result. So give me your take on it.
I mean, in terms of like kids getting into or young people getting into,
and especially in relation to like where the industry is going to be. Do you see enough opportunities out there for synthetic biology in the near future that it would be a good decision for people to go specifically and say, I want to do this type of synthetic biology versus like just get a broader degree or go into a more established field like, oncology or something like that?
Right. I guess I tend to tell people just get as broad of education as you can. Don't try to narrow it down. The more narrow of a degree you get, or the more narrow of experience you have, the less valuable you're going to be to the world, or the greater chance you're going to have of your field being obsolete next year.
That's a great point.
I think in the 90s, cybernetics was a big topic, and I think people who got a degree in cybernetics are having a hard time finding that job. And not because like they didn't have a good education, like they probably got a degree from like MIT or wherever, but now they have like this like totally obsolete term on their diploma. So I tend to tell people just like go to college, get a biology degree, get a chemistry degree, whatever. But don't like narrow yourself down into being like a synthetic biologist who only works on Saccharomyces cerevisiae. Just maybe next year that becomes not so cool, and now you're the guy with the weird degree and the weird specialty
that's of no use.
Yeah. And plus, it's having broader experiences is always good. Everyone says go travel after college to like experience the world. I wouldn't suggest traveling to like backpacking across Europe. I won't get much like cultural stuff. It won't help you scientifically.
But
in the same sense, like explore different fields that are interesting to you.
Very cool. I think it's sound advice, and it's advice I've heard from a lot of people. It makes a lot of sense. There's a lot of emphasis putting on specialization, but it seems that the people who come up with the most innovative ideas are the ones who make unusual connections, between fields or things like that. So a lot of opportunity there. All right. Well, just want to give people an opportunity to know where to follow along with the stuff you're doing. So Twitter, your handle there is LA Biohackers, at LA Biohackers. So, right. And then the website that they can view your work and kind of follow along. I found you guys on facebook.com/the lab, where the A in lab is a 4. You guys have a site aside from that?
Yeah. So the website is thelab.com, where the A is a 4. Unfortunately, the lab and like all permutations of that have been like taken a long time ago. So that's the unfortunate aspect of picking a name like the lab.
No, it's very fitting, right? As a synthetic biologist, to insert a 4 where the A is, I think it's genius. It's great. Well, Dr. Tobin, thanks for joining us on this. What it probably will be the first in a video series, but may end up becoming a podcast. I wanted to thank you for your time.
All right. Yeah. Thank you for having me. I look forward to seeing your videos.